I am delighted to welcome you to our conference focusing on the fascinating and valuable topic of regeneration in various conditions and processes. Our discussion centers on the significant role of stem cells in the human body and their importance in regeneration, which relies on both undifferentiated and stem cells. But what exactly are stem cells? Stem cells are formed after fertilization, resulting in a diploid zygote containing a complete set of chromosomes, from which all other cells that make up our tissues are developed, and ultimately, the entire organism. In the initial stages of human organism development, blastomeres, which are exact copies of the first stem cell formed by the combination of reproductive cells, become active. However, there is no involvement of the genome that would determine the organism's development, and there is no transcription in the nucleus. This lack of development in the nucleus indicates the immaturity of these cells and makes it uncertain whether they will develop or not. Therefore, our focus is on reflecting and exploring these undifferentiated and unproductive cells' potential. In summary, just like a test can either produce plants or cakes, stem cells blastomeres can develop into certain tissues, resulting in the development of specific parts of the human body. That's crucial to understand that tissue construction begins once a cell becomes specialized. For instance, mucosal cells can reconstruct to form connective, cartilage, bone, or nervous tissue. Defining specific body parts allows us to identify presumptive zones where neurons, mesoderm-derived blood cells, hepatocytes, or liver-forming cells will be constructed. Cell differentiation is acquired gradually through transcription experienced by stem cell nuclei. Stem cells are not only present during organism development but also after it is fully formed. Throughout our lives, we all have a set of stem cells in our bodies. With each differentiated cell having its specific function and life cycle, disruptions can cause congenital or acquired diseases. It's worth noting that the potential for stem cells decreases significantly in older adults. The primary properties of stem cells are their ability to regenerate without differentiation and produce specialized cells within limitations. Additionally, presumptive zones and stem cells' properties can differentiate into any part of the human body. As a result, stem cells are classified into groups depending on the age periods when present. In the embryonic section, these cells are pluripotent, enabling them to transform into cells of any tissue or organ, although they don't produce antigens that ensure tissue compatibility. Consequently, embryonic stem cells that don't cause rejection are widely used in therapies. A second group of fetal stem cells is currently under active debate and study. The capacity of these stem cells is limited, meaning that not all of them can be created or cultured in vitro. However, they are highly efficient and safe for use in therapy because they are autologous, which means they come from the patient's own body, minimizing the risk of rejection. Specifically, bone marrow cells activated in a specialized apparatus can be grown to the desired number before being reintroduced into the patient's bloodstream, initiating a natural regenerative process aided by the patient's own cells. Despite these benefits, there are still two significant issues related to these cells. First, there is no definitive research to prove, without a doubt, that these cells will not experience rejection or interfere with the recipient's immune system. While autologous cells should not cause a reaction, some bodies identify their cells as antigens due to failure in apoptosis. Furthermore, the fate of bone marrow-derived stem cells is still unclear, raising questions about the effects of culture conditions and the long-term presence of these cells in the body. Consequently, individuals that opt for treatment using these cells must sign waivers acknowledging the associated risks and absolving the doctors of their liability. Despite significant advances in stem cell research, we have yet to unlock the full potential of stem cells, and there are still many unanswered questions in this field. Nevertheless, certain groups of stem cells hold immense therapeutic potential, and there are even suggestions that they could enable us to live up to 250 years or more. Embryonic stem cells are universal and have the ability to differentiate into any type of tissue or organ in the human body. They are also specific, capable of creating various organs and systems. In contrast, postnatal stem cells are more limited in their differentiation potential, with hematopoietic stem cells being an excellent example. These stem cells can restore blood and are extensively used in bone marrow transplants for treating leukemia. Multipotent stem cells, known as MSC stem cells, are far more versatile, with the potential to regenerate a broad range of tissues and organs in the body. While specific stem cells are stromal elements, not all stromal elements are stem cells, and this variation is partly due to the idea of tissue specificity in which specific tissue cells restore a certain tissue that forms part of our bodies. Stem cells can also be obtained from placental blood, although the procedure remains fraught with some risks, with ongoing studies exploring the implications of this approach. Stem cells have, however, shown tremendous promise in treating over 70 illnesses, including cancer. As a result, they are stored in specialized banks, and donors can help save lives. Plant stem cells are less researched and potentially less dangerous than other types of stem cells, mainly because they contain less genetic information and have a more predictable effect on the human body. Plant stem cell-based creams have gained considerable popularity due to their excellent anti-aging effects, though the expense of such products is significant. It is worth noting that for decades we believed that nerve cells were unable to regenerate. However, groundbreaking research in the 1990s turned this understanding on its head, discovering that adult brain stem cells are the only active stem cells in the human body. Fascinatingly, these stem cells persistently retain their reproductive potential, giving rise to numerous progenitor cells. Brain stem cells possess an amazing ability to produce support cells for neurons called progenitor cells. These cells are responsible for the retrieval and transmission of nerve impulses and our reactions to sensory stimuli. Intriguingly, the precursor brain stem cells can trigger glial support cells production, which plays a crucial role in maintaining our nervous system during embryonic development and everyday life. These precursors are self-regenerative and produce offspring that are identical to the mother stem cell. As they are present throughout the body after development, they're referred to as somatic stem cells. They can reproduce through cellular division, replacing dead or damaged cells in our organs and tissues. It is noteworthy that these precursor cells in the brain can generate cells from any part of the body to regenerate tissues and organs. It's crucial, however, to mention that adult brain stem cells are restricted to a tiny region of the brain. Although our bodies produce neurons continuously, their reproduction process is slow. This makes stroke and brain injuries caused by tumors result in a swift preservation of bodily function and even death. Furthermore, discrepancies in the central nervous system's size can trigger genetic mutations that impair nerve stem cells and their adaptive potential. Stem cells in the nervous system must differentiate into distinct cell types, a process that can occur symmetrical or asymmetrical. While some of these cells turn into neurons, others transition into astro- or oligodendroglia cells in charge of our nerve microenvironment. Marine kelp algae have successfully promoted brain regeneration and slowed the aging process. Nevertheless, the tissue recovery process, even with nerve stem cells in the bloodstream, is often delayed. Thus, it's crucial to lead a healthy lifestyle to stimulate natural regeneration and protect our nerves from damage. The process of regenerating one's own tissue, including nerve stem cells, requires differentiation under the influence of specific humoral factors secreted by various tissues. Epidermal and hypokitic growth factors are examples of these factors for individuals experiencing stabilized symptoms, that is, when their condition enters remission and their state improves. The maintenance dosage should be at least 10 mg per kilogram of body weight. The dosage may reach 4 to 6 or even 9 grams per day during disease exacerbation, treatment, or onset, especially in cases of oncological processes. Each person requires a specific dosage. Children are particularly vulnerable to problems and require dosage based on weight and age. There is no risk of overdosing with ACTIVIDAN, even in small children. To administer an infant a packet per day is acceptable, but dividing the dosage into several meals is necessary. Each meal should have a specific dosage to ensure absorption by the intestine and prevent elimination. The timing of ACTIVIDAN use can be influenced by various factors, including age, tissue immaturity, and development. As a result, it is advised to dilute ACTIVIDAN instead of taking it in its dry form. To consume a packet of ACTIVIDAN, it's recommended to mix it with at least a glass of water. If administering fractional doses, more water should be added to ensure proper dilution. Nonetheless, in some cases where dry administration is necessary, ACTIVIDAN can still be used to treat certain conditions like stomatitis or bleeding, depending on the cause. For instance, periodontitis in smokers is an example of a condition that can be treated with ACTIVIDAN through applications. It's crucial to drink enough water after the tablet has dissolved to be continuously absorbed in the stomach. ACTIVIDAN serves as food for the body, supporting the regeneration processes and nourishing cells to optimize their functioning. Ensuring ACTIVIDAN is available for use is essential, just as one ensures water, air, and a healthy diet are always readily available. Many of us encourage others to be responsible for their health, including family and friends, highlighting the importance of taking care of oneself. ACTIVIDAN is an effective way to take care of one's health. There are no further questions towards the end of today's conference. It's hoped that attendees' curiosity has been satisfied and a better understanding of stem cells achieved. Nonetheless, the conference is not yet over, and attendees are encouraged to share suggestions for future conference topics. Eugenia and I are tired after many years, but we're interested in making attendees happy and surprising them at future conferences, so any specific requirements are welcomed. Until the next conference, best wishes.